Swayam Prabha. Digital India. Educated India.
Okay students, so today I will teach you a new topic on translational research that can be very much useful for nanobiosensing. So what is the translational research? So the main contents of the today's teaching, the translational research for the nanobiosensing. Especially you can think about the what is the definition of translational research first for today's lecture, and then nanomaterials for the healthcare and the biosensing. These two topics mainly I will cover today's slide. So, main keywords for this teaching for today is the translational research, that is the bench to bedside. The main definitions of the translational research, whatever we are doing in the lab, that can be useful for the point of care diagnosis, that is the near the patients, that is called the bedsides. So, that I will show you in detailed way. Then nanomaterials for fabrications of the biosensors and the detections, and how we can use the translational research for nanobiosensing. See, the translational research is very unique topic. It is the thing is the laboratory breakthrough to the patient care. Translational research, the main thing, it can expedite the discovery of the new diagnostic tools that can use the multidisciplinary technique. So, why translational research is needed? Because it can expedite the discovery. Now, let us come to the translational research first. So, you can see in the image in the left side in this slide that at first we are doing different kind of research in the laboratory. Then we are going for animal trial and we are validating our concept. Now, we are transferring this idea to human trial for applications. Maybe we are developing some drug or developing some new therapy that we have to try for human trial. Before that, we have to optimize all the conditions in the lab. You can see in this left hand side here, actually we are first doing the laboratory testings. Now, we are transferring this idea to human trial. Now, we are giving to the doctors and they are trying this drug or your technique. Suppose we are developing some detections method. So, we have to, after the validation, you have to give to the doctors, that they will validate it. And it is not the unidirectional method, it is actually the both directions. Once we developed in the lab and we are giving to the doctors, they may find some problem. It may not be useful, there may be some side effect, or maybe we can improve this technique. It is useful, but may not be perfect. So, what we can do? We can come back again to the laboratory again, and we can improve and we can try to recover the issues. So, again we can come back to the lab, we can do the testings again, we can again go for the animal trial, and again we can give to the doctors or to the hospitals, they can try again, they can validate your technique like this way. So, like this way only we can expedite the your technology, your development, and that can be useful for point of care. So, that I taught you the nanobiosensing technology, that is same thing. What we are doing in the laboratory, we have to go for the real sample testings, then we can ask the doctors to use these, and if they find it good, then we can go for commercialization. So, as that's why I am saying it is very much multidisciplinary and is the highly collaborative. Means basic scientist, we are doing the research in the lab, then we are giving this one to the doctors. So, we need the always the collaborations with the clinicians. Okay. Now, before going to the nanobiosensing translational health research, very briefly I will tell you some models of the translational health research. There is some models like T0, T1, T2, T3, T4. So, T0 is the very basic stuff that we are doing inside the lab, that is a very basic science. Then we are going to the T1 and T2 and this highest phase, that is the clinical trial, and the finally the medical applications. Means first we will try in a small group of the members, maybe in the after the animal trial to the small group of members to the human trial, then we can get the large members, then we can spread this one to not only the maybe suppose we are thinking the local region to the nationally, then we can spread to the like internationally. I mean how much effective in the region wise your technique. So, there is a different technique, that is why. So, this is the very preliminary, then translational human research to a small group to the higher group, and then we are trying this technique to validate in the all over the region. So, our main goal of the translational research, that is why you validate your technique for the mankind. So, why translational health research is needed? Because there is some current issues, because whatever we are developing in the therapeutic or the medicine, for example, in the cancer, right. So, we are using the ionizations, radiations, but there is some drawbacks. What kind of drawbacks? Like there can be the secondary cancers possible, there is skeletal complications possible, there is radiation induced heart disease, lung disease. So, we have to improve the technique, right. Another things like chemotherapy case, I think you already heard about this, that chemotherapy it is not the very easy technique, it is highly sometimes toxic, damage the normal cells, allows the cancer cell is damaging, at the side by side can damage the normal cells also. There is some secondary cancer also may be possible, and some chemotherapy drugs delivered through the solvent that are not easily taken up by the body. So, we need the further improvement, right. So, that is another things I will show you this image, that they are possible by the tumor regrowth, also insufficient primary therapy. So, this kind of problem there is a risk can be recurrence again, like because the therapy or drug we developed sometimes different person to person, it is not be useful for everyone. So, we need some further development, that is why. I will show you one image here. You see generally what we are thinking, like a cancer patients we can go for the therapy. So, we are giving some medicine. So, it may be useful for certain group of the people. So, it is very much effective, but some group case there is no effect, but some group of the people they may be adverse effect. So, what translational research can do? You can go for further study. What you can do? You can go for blood, DNA, RNA testing. Then we can divide the people like certain group. So, then we can apply some certain therapy for the different group. So, it is the more personalized we can develop, and this approach can be more successful. So, you understand, right, the translational health research is very simply and very briefly I am saying, because it is this course is not for the translational research, but this concept is useful for biosensing. So, main theme is the bench to bedside that we are planning. So, let us come to that. So, now nanotechnology, the nanobiosensing, that is I told. So, it is very innovative technique, and we can easily integrate this one with using different kind of nanomaterial. So, few years before we were thinking it is kind of the impossible fantasy, but now we are really using this nanotechnology for current drug development, for current like therapy, and there is lots of future perspectives up there. You see that for the drug delivery is also the very important, and it can optimize the drug efficiency, and it can reduce the toxic effect if we can use this kind of the nanomaterial based. So, these bench to bedsides applications using the nano technology is really it has a future perspective. We can use this one. Now, let us come back this translational health research, how we can use for nanobiosensing. It is not only the nanobiosensing, like any kind of medical innovations. See, I will give you one example, that is the study of malaria. So, see, the malaria is a life threatening disease that mainly infected by the mosquito carry the Plasmodium parasite. So, we can think about the bench to bedside applications, like we can develop a sensing technology, so that we can detect the malaria in the early stage and we can go for further treatment. So, doctor should know that it is because of malaria or not. If you come to the doctor with fever, so people should detect the it is because of malaria or not. So, what we are doing? So, first we can develop some sensing method. You can see here in these pictures, actually we collaboratively work with Professor Peter Lillehals in the Michigan State University. So, where we developed a mobile phone based smart nanobiosensors that can detect the malaria. See, when we use this kind of mobile phone based sensor for detection of malaria, we tried in the African countries and there, and we developed some sensors that was beautifully worked in lab, but when we went for the trial, we faced some issues. We are developing so many techniques, but you may plan, okay, it is very much useful for the patients, it is very sensitive, we can use for the point of care, it is absolutely fine, but maybe there is some another requirement for the field testing, right. So, I can show you the requirement now. So, we first went for the testings. So, Professor Peter Lillehals group, they went for testing this one. Then they found actually PF-HRP2 level is for the uncomplicated malaria progress to cerebral malaria is significantly higher concentrations of the PF-HRP2 level. PF-HRP2 is a biomarker that cause the malaria. So, we can detect this biomarker if you have a malaria. So, we found that this marker is very high concentrations in the blood samples, and it can go up to 90,000 nanogram per ml. But whatever biosensors we developed, that is very sensitive, it can detect like picogram to nanogram level, but it cannot detect like 90,000 nanogram per ml. So, our sensor now failed because it becomes it's saturated, it cannot detect the very high concentrations. It can detect very low concentration, not very high concentration, but in the case of uncomplicated case, this concentration is very high. So, then how we can use your sensor? So, it is really a problem. That problem we found when we went for the field testing. Now, what we did? So, we came back to the lab, then we developed the another sensors, very simple sensors. It is a paper based sensors. After a few class, I will teach you this kind all the electrochemicals, all the this kind of like reference, what is reference electrode, working counter electrode, that I will teach you later. But here you can see a very simple sensor we designed for this malaria detections, where we use the blood samples and different paper membrane we used, and we use some bioreceptors of the antibody on the sensor surface that can specifically detect the PF-HRP2, and it will not be saturated easily. It can detect very high concentration. So, we use some technology here, that technology I will describe you another day, but we developed some new technology that can detect the PF-HRP2 in blood sample in a very high concentration. You can see here that we published from we published, I worked with the Professor Peter Lillehals group. So, there we published that scientific report. Here you can see that very high concentration with the microgram per ml, like 100 microgram per level, in this much concentrations we can detect the malaria. See, so then we solve this issue in the lab, that whatever like picogram, like very small concentrations to high concentration, now we can detect. So, this sensor is ready now for again field testing. So, see, that is why we put here, see this both directions, like first we tried in the lab, whatever you did your technology, then you went for the testings for filter, then we faced the issues like your sensor is not applicable because it cannot detect high concentrations, then we came back to the lab again. Then we developed again a sensors that can detect the very high concentrations of the biomarker. Now, this sensor is ready. So, we can now commercialize this kind of technology. Okay. So, this is very like important technique for nanobiosensing also. Okay. So, there is you should read some reference papers also, that I told you that I already referred some journals. Also you can investigate, like this kind of the papers is really important. You please check it, and you will get lots of more information for the translational research. And this is not only for biosensing, for different kind of like cancers and other disease marker detections, and not only the like simple device development. You can go for like therapy, or like in nanotechnology based some drug treatment, that also you can check. So, the conclusions of this story, that is why the translational health research is very, very important, and I will show you one image here. See, so this image you can clearly see our main innovations, practice and research. That is why it stands with the two very important strong pillar. One is translational research, one is implementation science. So, we have to implement our research. So, based on this translational research, and this main base is the collaborations. So, only basic scientists cannot do. But see, so only like one chemist or one biologist, they can develop very sensor, very simple technique they can tell you, like antibody, they can detect the some specific like PFHRP2 antigen, it can detect by the help of like maybe you can develop some sandwich method by ELISA. That basic biologist that can tell, it can tell you, but you have to translate this technology to a sensor surface, so that one engineers who are developing the sensing technology, he can help you, like he can design or he can pattern a sensor on a like nanomaterial coated but on the gold surface or any kind of like different carbon surface. So, he can pattern for you, then you can translate this very basic concept on the sensor pattern surface, right. So, now you checked this sensor is working. Now, you have to collaborate with a device maker, like who are developing the device. Maybe you have to collaborate with the electronic background peoples also, because whatever signals you are getting from because of these reactions, and you are getting some electron transfer because of this reaction. So, you have to collect those electron and you have to amplify those signal, and you have to show this signal to a display board. So, you need some electrical circuit, some display, like a mobile phone based some technology you need. So, some electronic people, they can help you. So, you need basically the collaboration with them. Then once your sensor ready, then you have to collaborate with some medical doctors, right, the clinicians, they will help you for validations, right. So, that is why I said this translational research is very important, and it is very much collaborative approach. Then only your innovation can be successful with collaboration with other group. So, we can go for the collaborations and the team science for complete development of your innovations, and translational health research also can be useful in the complex medical systems. So, not only that this very simple technique, even the complex diagnostic technique, complex therapeutic technique, sometimes that is why development of a certain drug can take 10 to 12 years. Why it takes too long time? Because of this, because first you are developing some medicines, then you are going for trial, and then finally you are validating. So, it takes something too long time, but these translational research also can reduce the time. Then translational research, it is a very advanced study design. This translational research is very, very advanced technique, and nanoscience for translational research can help you for further improvement. Also, it can help to reduce the time, this nanoscience. Okay, so that is all for this translational research. In the next topic, now I will show you the different biosensor based on nanomaterial, and that can be useful for the different signal amplification and the biosensor development. Thank you all.